All right, so today's lesson focus is actually on you being able to practice doing transcription and translation. So I'm going to quickly run over how to do both processes, and then I'm going to have you actually practice and then do your STAR questions in your interactive notebook for today. So again, the learning objective is similar to the ones we've had before, where students will be able to practice the processes of transcription and translation to construct polypeptide proteins starting from DNA sequences. And remember, we want to answer the questions, why can't the DNA directly make the protein? Well, DNA is only instructions, right? It doesn't include the machinery to make proteins. And the machinery to make proteins is in the cytoplasm, and DNA is in the nucleus. So it's going to have to send messages out to the cytoplasm from the nucleus because it can't leave. That's why DNA can't directly make the protein. It can't leave the nucleus. And we also want to think about, how are the 4 nitrogen bases of mRNA different from the 4 of DNA? Well, there's only one actual difference. Adenine A, cytosine C, and guanine G are the same for mRNA and for DNA. The only difference is that DNA has thymine, which is represented by a T, whereas mRNA or any RNA has uracil, which is represented by a U. They substitute for each other in the same exact place. So in RNA, U and A pair together, and then in DNA, A and T pair together. So functionally, they're the same, but you have to recognize that when you see a U, it means that it's mRNA or RNA. And if you see a T, it automatically means that it's DNA. So let's just basically go over transcription again. It's the process of turning DNA into mRNA code, because the M stands for a messenger, and we know that DNA can't leave the nucleus. So the mRNA message is going to leave the nucleus after it's made and go to the cytoplasm, where then it can be converted and used in the process of translation to make the actual amino acid protein physical product of the central dogma. An example that we're going to do right here is that is related to the rules I just stated on the earlier slide, where the pairing rules for RNA are that A pairs with U, that C and G pair together. But since you're starting with DNA, you'll see the T in the DNA, and the T always pairs with an A when you're making the mRNA. So let me do some examples right now. So if you see an A in DNA, you're always going to pair it with a U in mRNA. If you see a G, you'll always pair it with a C. If you see a G again, you're going to pair it with a C. Now, the reason why the G's and C's are always paired together is that's just how they match up in any kind of nucleic acid. The A we paired with a U because it's not going to be paired with a T since it is in RNA. If we were making DNA to DNA, like in DNA replication, then we would put a T. So now in this next codon, we see a C, so we're going to put a G. We see an A, so again, we're going to put a U, and then we see a T, so we're going to put an A. In this next codon, we see a G, so we'll put a C. There is a C, so we'll put a G, and there's a G, so we'll put a C. In the next codon, we see a T, so we'll put an A. We see another T, so we'll put an A, and then we see an A, so we'll put a U. In the last codon, we see an A, so we'll put a U. We have a C, so we'll put a G, and then we have another A as the end, so we'll put a U. And that makes the mRNA strand UCC-GUA, CGC, AAU, UGU out of the DNA strand. This is the process of transcription. So whenever you see an A, you're going to pair it with a U in the mRNA. Whenever you see a G, you'll put a C. Whenever you have a C, you'll put a G. And then whenever you have a T, you'll put an A. And that's always what you do from going from DNA to going to mRNA. Now, we're going to take this exact strand that we just made in mRNA, and we're going to do the process of translation with it. And translation is a little bit more complicated because the transcription rules are pretty simple. It's just when you see this, you put this. Translation requires you to look at each codon individually. Now, it's the process of turning mRNA code into amino acid sequence, which is the actual protein. Anticodons on tRNA match up with the codons on mRNA to bring in specific amino acids. Now, I don't want you to forget that codons are triplets of nitrogenous bases. So the reason why I've grouped them into 3 at a time here, like UCC, GUA, CGC, because those are codons. They are 3 bases at a time on the mRNA. Anticodons are obviously the exact mirror opposite that match together on the tRNA. If it doesn't match, it's not going to bring in that amino acid into the protein sequence. So again, a codon is 3 at a time. So we're going to be bunching 3 at a time, which is why I put the dashes in between. It helps you visually separate out one codon from the next codon. It makes it easier to read to find your amino acid. And this process for translation happens in the cytoplasm on the ribosomes, which are the protein making factories. The example we have here is the same exact strand that we made on the last slide, but when you're doing translation, you actually need this universal genetic codon chart, which uses mRNA or messenger RNA codons and the amino acids that you will be putting down. And if you can see on the side here, there's this first base, second base, and third base. And this is really similar to the one that you will be using on your STAR questions and on any of your tests. But the way it works is that, for example, you're going to take the first codon, which is UCC, and we're going to take the first base, which is U, we're going to take the second base C, and the third base C, and find out what amino acid will belong in the matching section. So the first base, like we said, is a U, which means we're going to be looking in the U row. You'll find U, C, A, G. So we'll look in the row that actually is represented by U. The second base is up here. We want to look at C because the second base was a C. So in this row, there is one box that represents C as the second, and that would be over here. Lastly, we're going to look at the last base. So we have UCC, and inside of this box right here, we see that the second one says UCC. So our amino acid that we're going to write shorthand down is going to be SER. So we're going to write SER here. And we're going to move on to the next codon. So the next codon is GUA. We're going to look on our table, and our first base is G, which means that the row that we're going to look at is not going to be U, not going to be C, not going to be A. It's actually going to be G done here. So we have GUA. So on second base over here, we want to look which one is going to say U. And that actually happens to be this column. So we have G first and then U second. We're going to be in this box. And you want to look for GUA within this box, which will be down here. And we know that the shorthand for this amino acid for valine is going to be VAL. We're going to write a VAL. Okay, the next codon we're looking at is CGC. So we're going to go down here, and our first base is going to be C. Our next second base is going to be G. So we're going to be looking in this box because we know that this box has the C as the first base and the G as the second base. So here we're going to be looking at CGC. So inside of this box, we want to find CGC, which is right here, which is going to be ARG or arginine. And we're going to write ARG as the shorthand. Now, the next one we're going to look at is AAU. So our first base is an A. Our second base is also an A. So we're going to be in this box. And AAU is shorthand represented by ASN. So we're going to write an ASN. Our last codon is going to be UGU. So UGU on the mRNA codon chart is firstly U, second base G, last base U, which is right here. Okay, again, first base is U, second base is G, and they cross each other in this box. And inside of that box, we're looking for the last base, which is UGU, and we'll find that UGU is shorthanded by CYS for cis. And that would be our amino acid sequence that came out of this mRNA strand, which originally came from the DNA strand. So with that being said, that is the protein sequence that came out of the DNA. So you had your instructions, you sent your message, and now you have your protein product, which is the sequence of ser, val, arg, asin, and cis, which is which represents shorthand for a specific order of amino acids in protein. Okay. You're actually going to take this example now, and you're going to practice on your own doing the central dogma practice, and then you will do some STAR multiple choice questions in your interactive notebook before you finish out with your exit ticket test.